And we're back. All right. I hope that was great for everyone. Thank you to the presenters from the first bit. Now we're moving on to the second part, and Abby's going to lead the way with a thesis in neuroscience. So all the best. Daniel was next.
What would you do if I told you that you only had 15 months left to live? That's the reality for anyone diagnosed with glioblastoma brain cancer. That was the reality for my dad. There's no cure right now, and so we need more information and research. Our understanding of glioblastoma formation is a relatively simple formula. If you direct your attention to the top of the screen, human plus mutation in the brain results in a tumor. Tumor wants to grow, and it hijacks the blood vessels for nutrients. Now, studying glioblastoma, that's a little bit harder. It would be unethical for me to say, I'm going to give you a brain tumor now for the sake of science. I hope that's okay. So instead, we use models where we can ethically produce brain tumors and cut them open and see what's happening inside. One of these models is the fruit fly. Fruit flies have been favored by geneticists for the reasons that Jenny stated. They share a lot of DNA with humans, and they're really easy to genetically modify. So easy to modify that we can give them the very mutation that causes brain cancer. How? Imagine two genetically modified flies. You can think of these like a map to a treasure chest and a key to unlock that treasure chest. The first fly, the map, has the directions on where to go. In my case, it would be to special cells of the brain called the glia. The other fly has the instructions on what to do there, like introduce a cancerous mutation. You breed these two together, you're going to have offspring with brain tumors. Cancer in flies might look a little different, but it follows the same formula. Fly plus mutation results in tumor. Tumor grows by hijacking the vasculature. The problem is that no one has put these two pieces together in fruit flies. And so that's what my thesis sought to do. First, I established a baseline of normal brains in fruit flies, cut them open, dissected them, and I imaged them looking specifically at those glia cells and the vessels. Then I gave the flies brain cancer using the method I explained, and I cut them open and imaged them the exact same way. All that was left was comparing across the two groups. And I found that my cancerous fly line was significantly different from the non-mutated fly line, which means that my study was the first step in establishing a consistent methodology to model these two elements of brain tumor progression in fruit flies. It gives me hope that in the future, brain cancer won't be so deadly a diagnosis, that my research will be able to contribute to that, and I can offer someone just a little bit more time with a loved one than I was offered with my dad. Thank you.